I knew at the end of a long meeting where our clinical community would be sitting here hearing stories about how real patients were treated in real hospitals around the UK, that even our persons might be wondering what the basic scientist has got to offer to these symposia. And I think Elaine has started by already doing a job of anticipating exactly what fundamental research does to move things forward. So for Brian and for Peter, if you are not uplifted enough by the experiences of the last hour, these are some of the reasons why I think that some of our work and some of the other work you're going to hear about, why it contributes to this debate and this update of the state of the nation in medical mycology. So clearly, people are interested in this symposium about the use of diagnostics. We had that fabulous session yesterday, and we've heard about things like MIC testing, and there is a kind of supposition, of course, there that for those to work, that it would be very convenient if the target that we were chasing down didn't change its properties too much. So one of the things I want to reflect on is the reality is that the things that we are trying to deal with, these medically important fungi, are exquisitely variable organisms. And we've began to hear a little bit about immunotherapies, but what is the consequence of the treatment of a fungus with an antifungal drug in terms of its immunological signature? That's another issue which we need to think about, because you've already heard again at the end of the last session that the way in which a fungus causes disease is not simply to do with its virulence properties, it's how it manipulates the immune system. And what are the futures of immunotherapy? Tom raised this issue today. Challenging, but clearly desirable. We want something different to offer in the future. So if you're looking at a fungus, what are the bits of the fungus which do these jobs, which influence the immune system? And that's really been the focus of my group's work. And then you should also keep awake, because we're anticipating in an update what might happen into the future. And the use of traditional methodologies along with developmental methodologies is clearly going to be important. So that's why I'm keeping an eye on you guys to see if you can keep your eyes open. No, it's not working. So let me start with a positive example. I'll choose this example from one of the group working in Aberdeen, Gordon Brown's group, of a piece of observation, a new one, relatively new one, something which happened in the last few years, which is already reaching a clinical trial in the context of a specific disease. And this is, I need to remind you for this, about a slide, and this is almost exactly the same image you're seeing here as Elaine showed on hers. It comes from the same review. And this point of this graph is showing that when you look at these pathologies generated by fungi, that you're looking at two forces at work. Sometimes you're looking at the properties of the fungus causing disease, necrosis, tissue destruction, etc. But other times, when you have a very strong immune response, you're looking at the immunopathology of the overactivated inflammatory response. And actually, fungi do both of these things. So you really need to know in what context that you're looking at a particular organism and its disease setting. But the one I've chosen, the example, is Gordon Brown's work on chromoblastomycosis caused by a number of organisms, of which the most common is this Fungal petrosii. It's a particularly nasty-looking disease, very chronic disease, skin infections which are very recalcitrant to chemotherapy. It often requires surgical treatment as well as long-term chemotherapy. And what Gordon noticed is, he's an immunologist, as many of you will know him, here he is here. He's interested in, and he's the discoverer of Dectin-1, and he noted that for Fungal petrosii to be recognized by the immune system, it required multiple recognition mechanisms. In fact, Dectin-1 doesn't play a particular big role here, but the other C-type lectin, Minkle, does. And he knew that if you have one recognition mechanism, this red one doesn't work as well, but green will work, then that doesn't kick in the immune system, the protective immune system, enough. So he reasoned there, well, what happened if I used an adjunctive therapy and boosted it with something which synergized with the C-type lectin, two recognition mechanisms, and he just used simply bacterial lipopolysaccharide. And whether it was injected intraperitoneally or IV, he could actually eliminate the fungus in a mouse model. And this has now progressed to a clinical trial simply by using a cream with a TLR agonist as part of the therapy, and actually it's giving very, very positive results. So there is hope that basic findings made by laboratory-based studies might pave the way for novel therapies into the future. Now that work relates to recognition of the fungus, and this is what my group mainly works on, the cell wall. And I think there's a lot of reasons I can justify to my own staff why we should be there, working on the cell wall. The cell wall really of a fungus is its signature and its life. It's its protective outer surface, and it's the way in which it communicates with its world. And in medically important fungi, that's us. These are the sort of physical properties which are conferred by the fungus: its shape, its strength, its elasticity, porosity, etc. All of its biochemical properties. It's also, of course, a target for antifungal drugs, and it really dominates the immunological signature, both in terms of what activates the immune system and turns it off, and which modulates the immune system. So by studying the fungal cell wall, you gain a lot of traction and a lot of fundamental problems, and that's why we're in that space. And in fact, about a fifth of most fungi devote their genome to assembling and then maintaining their cell walls. And these cell walls, we used to think of them, it used to be a rather dull area of cell biology. People used to think of the cell walls as like an eggshell, rather an inert structure, hard and robust, but nothing more like that. We know that's not the case anymore. It's like a highly regulated organelle, and it's exquisitely responsive to environmental changes. So although it's the target of antibiotics and of immune detection mechanism, it's also a moving target, a highly variable target. And I think that's something which is very important, both fundamentally and clinically, to be aware of. So, for example, under various conditions, a fungus is exposed to different micronutrients. It has to grow on something, and for Candida albicans, which is in this cartoon here, most of the time it's probably living on glucose concentrations within the serum and the blood and other tissues. But in necrotic tissues, there's very little glucose, and it probably lives on things like lactate and fatty acids. And as it turns out, that significantly changes the physiology of the fungus in all the sorts of properties that you as clinicians would care about. And I want to show you some work led by Al Brown at Aberdeen to justify this. Now, unfortunately, the little blast of sunshine is taking away a key image here. But here on the top left of this panel are two pictures of the cell wall of Candida, which is grown on lactate as a carbon source and glucose as a carbon source. And you probably can't quite see it in the glare, but in the lactate-grown cells are completely different in image. They're much, much thinner, about half the thickness of a glucose-grown cell, but actually they're very tough. Despite the fact they're thinner, they're actually much more robust and more stress-resistant than a glucose-growing culture. And if you look at, say, the proteome of the cell, and it's just the blue bars compared with the red bars, again, the proteins in the cell wall are completely different on those two conditions. So you have one fungus which is moving between different microniches within the body, and it's at a completely different physiology. Actually, the sort of physiological differences that you see here are about as different as you see between different species of Candida. And if you look at any other type of readout, for example, these two graphs, the dark bars and the light bars, are for two different cytokines. You can see they're very, very different. And down here, bottom left, are virulence assays for glucose and lactate-grown cells. And they're more than double the difference in their relative virulence, depending on the carbon source at which it's growing. So this is a very movable target. It affects also things like drug-resistant sensitivity and all the things that we care about in terms of trying to deal with fungi as medical problems. Now, the second thing I want to turn to, which is the main theme of my talk, is that this variability is expressed in the surface components of the cell. Now, all of the bits of the cell wall, fungal cell wall, are pretty much specific to fungi. Most of those molecules, almost all of the molecules, It would be not an oversimplification just to say they're all completely different to the molecules you find in human cells. So this is a fantastic reason why the evolution has recognized the fungus to target it as being an invasive pathogen, but it's also a very good opportunity for people who are interested in designing molecules to kill fungi. Now, what it turns out is that these various components of the cell wall are dominated by sugars. There's a mannan outer layer, and that's that fibrillar layer there. And then there's this amorphous layer, which contains a little bit of chitin, a lot of which I'll tell you about a bit later, quite a lot of beta-glucan, and linker molecules of beta-1,6-glucan. And all of these molecules tend to be, we now know, are activators of the inflammatory response to various extents, and I'll show you some of the evidence for that in a second. The proteins themselves, which are these blue molecules here, are very variable as well. The most regulated genes in the Candida genome are the proteins, are those that encode proteins which go into the wall. Now, can I find the cursor? This, where's it gone? This, for example, is a one protein of the cell surface, one biomarker if you want the cell surface. It's a bit like Chris Thornton's one because it's only found at the hyphal tip, so that's a heat map image. And this is an antibody which is part of another story which I won't talk about today, which is generated by a fantastic PhD student in the lab, Fiona Rodkin, working with myself and Lars Erwig. And she's actually generated these antibodies by a very novel methodology. She's actually cloned the genes for the antibodies directly from individual single B cells. And this is a very powerful mechanism for generating antibodies because the antibody you get is a completely human antibody. So you don't need to re-engineer it to be compatible as a drug to inject in the body. And it works very well. We've raised them to specific proteins and also to whole cell surfaces. And that's a very variable component. So you have these constant components and these variable components. But even these components are actually vary quite a lot between individual Candida species and within one species, which has grown in different ways. Now, the way in which the immune system recognizes a fungus is through an array of receptors of several different categories, lectin-like receptors, toll-like receptors, and other types of cytoplasmic receptors. And predominantly, you can see at the top here, they recognize carbohydrates. And most of them, in fact, recognize mannans. And mannans are not very common sugar in the human body. So most of them take advantage of the fact that fungi have a lot of mannose-based sugars on their cell surface. And of course, it's very interesting and important to know what exactly these individual receptors recognize, because for many years, immunologically, people have just said, well, they recognize mannans. But mannans are not one molecule. It's like saying they recognize proteins. Well, what protein do you mean? What exactly do you mean by that? So a lot of the work in our group has been focused on trying to understand with more molecular precision exactly what a fungus actually sees. And the way we've done that is by generating a library of mutants, which are like molecular scissors, which clip away specific epitopes, different sugar residues, and then you can say, well, how has the immune response been altered? And this is just a series of images of different glycosylation mutants of this type, and this is where the lesions occur in this cartoon. And we know that this is relevant because we can, with our mutants, we can very clearly depict the sequence of events which leads to the recognition of a fungus. So, for example, this is a set of relatively recent work by Becky Hall, who made a single strain which actually in the end had 12 mutations. It was a real tour de force to do this. She deleted an entire gene family which elaborated the N-mannan structure, the dominant N-mannan structure of the fungus. And as she was assembling this mutant, you can see this very hairy cell wall. It went through a series of haircuts until it became very bald, and you can see it was just almost devoid of its manna protein microfibrils here. And then when she put back genes successfully, she could start to recover some growth, some Rogaine-like experiment here to regenerate hair growth. And when she saw that, so here's the length of the fibrils here, and what happened in the series of experiments in terms of immune recognition is you saw that the balance of power in terms of recognizing the fungus was transferred in the wild type from recognizing mannose-based sugars to, in the bald mutant, to recognizing beta-glucan, the underlying layer, via dectin-1. So that's very reassuring. And then some even earlier work showed that if you understood all the signatures, which collectively said this is a Candida albicans, you could essentially eliminate them all and get a fungus which couldn't be detected at all by the immune system. And that's shown in this image here. So here's 100% recognition, and this is what happens when you eliminate recognition of N-mannan. You get a big loss of recognition signal, and then when you eliminate recognition of O-linked mannans, you get a further loss. And then if you eliminate the dectin-mediated beta-glucan signal, you're down to about 2% of the recognition mechanisms. You can get an immunologically invisible fungus. So that tells you that these are the molecules that evolution has generated to be able to detect a fungus and act on it. Now, out of this, and this is where the last part of my talk is going to be focused, when we were doing this, I was constantly frustrated because actually much of the work in my lab for many years had been focused on a molecule I haven't yet really mentioned much, but it's a real signature molecule for a fungus. Actually, every fungal pathogen has this molecule, and that's chitin. And we had very little understanding of what chitin did. There was all this work on beta-glucans and detection by dectin-1, and almost nothing on chitin. And I thought, well, we have to try and do something about that, because this is a very important molecule, and it might be just as important as something like beta-glucan in terms of immune recognition. Now, chitin's a very interesting molecule. Nature's been around for several billions of years, and this is the toughest thing it's ever come up with. Chitin is stronger weight for weight than stainless steel. It's a very, very robust molecule. A little goes a very long way. And you can abuse it very badly. What it is a single, it's a polysaccharide made of one sugar, but it folds back on itself and hydrogen bonds to make these beautiful microfibrils, which can survive even boiling an acid and alkali, and then you can image them in an electron microscope. And interestingly enough, although that's only one sugar in one bond, the chitin architecture in different fungi is quite different from one another. So there's Candida, which is like little crystals, and these are other filamentous fungi, which is much more like a network or a lattice. So they're molded together in slightly different ways, but nonetheless, it's a common denominator for all pathogens. They only have a single sugar. All of these preparations have just got N-acetylglucosamine in them. So what does it do? Well, the lead into this actually came from a different project in the lab. And it was when a group of us, including Carol Monroe and myself, were studying the effects of the fungus to respond to Casper fungin. Now, as you know, Casper fungin is something which damages and attacks beta-1,3-linked glucans. And what had been in the literature, in Saccharomyces, had been the observation that when a fungus is under attack, the cell wall is being compromised, particularly if beta-glucans compromised, what it does is it makes more chitin. And it does that as a mechanism to protect the cell wall from lysis. It's probably evolved that mechanism over hundreds of millions of years because fungi have been gobbled up by phagocytes in the soil, the amoebae, etc. It's a very ancient mechanism, a protective mechanism to store cell wall robustness. So you see a thickening here of the brown line. And what we wanted to know was, well, does that happen in response in a clinical situation? And what would be the other consequences of this? And there's several publications of this, but just to point to the important data, is here's an MIC test against Casper fungin for one clinical isolate, and you can see that there's good control of growth over all of these concentration ranges. And this is exactly the same strain, not a mutant strain. This is the same strain, but it had been just grown in a way which elevates its chitin content. You can do that simply by culturing in different media. And it has actually 7 doubling dilutions difference in its MIC, less sensitivity. So in a couple of hours, this fungus can go from an MIC, which is right down here, totally sensitive, to one which is far more resistant. And of course, we were interested in whether that was something which would influence the efficacy of these drugs in a clinical setting. Now, I can show you that if you're not convinced by those data, by just showing you what happens in a little movie when you expose Candida to a kind of candida, Casper fungin, and just follow it through a few generations. And although it's a little bit bright here, I think you can see the amount of blue fluorescence, which is chalcofluorescence, indicating chitin content, increasing very dramatically. And if I show you this movie for Aspergillus, you can see it's a very similar. Oh, wait a minute. Let's go back to find my cursor. If I run this in the bright field phase, you can see that the initial response is lysis of the tips, but then if you wait a bit, there are surviving portions of the mycelium which grow and form these rather gnarled-looking thick hyphae, which nonetheless hyphae which are continually able to grow even in the presence of that same concentration of Casper fungin. And if I now show you that same movie, but in the fluorescence channel, you'll see that nothing to begin with, because it's too bright here, but eventually you will see those regions which do grow are growing and they have high levels of fluorescence. I don't know if I've actually activated it. No. Yes, it's going. And the surviving parts of the mycelium are very chitin-rich. So what we knew is that these fungi, and that's Candida species and other species, have this common response to things which damage their cell walls by trying to protect themselves by making a lot more chitin. Now, as it's turned out, we of course wanted to know whether this had any in vivo relevance at all. These were entirely in vitro experiments. So we did this mouse model with Donna McCallum and Kathy Lee, and we used cells of different chitin content, low chitin content or higher chitin content, and we put them into mouse, and this is what we got. And what we could see here, so if you compare that bar, that blue bar, with this red bar, and these are kidney burdens in colony-forming units per gram, that these bars are the same height. This means that even in the presence of Casper fungin, we had the same kidney burden for the cells which had this higher chitin content. And that told us that yes, that those cells could be protected by virtue of having a higher chitin content, and the fungus was alive. But the curious thing about this experiment, and this is what the link is to the immunology, is that we noted that the mice were also alive. So if I show you the death curves here, it's the same color code. These are the data with the dark blue lines and the dark blue spots, showing what those of you who use mouse models will know that typically in a challenge model, like an IV challenge model, like a tail vein model, it'll be a couple of weeks and the mice will succumb. And this is the same fungus again, not a different fungus, not a mutant fungus, just one with a higher chitin content. Yet we lose 20% of the mice, but then everything else is stable, and those mice look quite normal, even though their kidneys are packed with Candida. That was a mystery. What was going on? So the next thing, we remember this. The pathology in these situations is driven by inflammation. Remember that. What we didn't know was what were the receptors. We had to find that out to make a guess about what was going on. And the first thing we knew is, very unusually, when these fungi are exposed to an echinocandin, the chitin which is built to protect the fungus appears not where it normally appears, in the deep wall, but is presented at the surface of the cell. And we knew that because this stain, which is a different chitin stain, it's a lectin, which is a very high molecular weight glycoprotein, wheat germ agglutinin, that doesn't percolate into the deep layer, and it will only stain molecules which are on the surface. So it doesn't stain well-type cells at all, but it does stain Casper fungin-treated cells very well. And we know this by EM and other reasons now, other methods now, that the cells which have a lot of chitin on the surface, the chitin which is produced, the extra chitin which is produced, most of it goes to the surface. And the next thing we found out was actually those cells were less inflammatory than the cells which had normal levels of chitin. Not only that, and this is the most recent data set, Jeanette Wagner, who's up here, she followed up on this and showed that the dominant cytokine which came from these high chitin cells was IL-10, an anti-inflammatory cytokine. And she used this as a tool to try and find out what the receptors were. So the way she did this, well, we struggled for a long time, I can tell you. But the best way forward was simply to screen a whole series of mutants we got from our collaborators and friends which lacked various receptors, and said, what happens to the IL-10 response? And you can see here, so that's an IL-10 readout here, and you can see there's flat lines for here. And these flat lines depict this pathway, a lectin-like receptor on the cell surface, the mannose receptor, and two intracellular receptors, TLR9 and NOD2, and a signaling pathway under here. And again, in this light, it's not very convincing, but there is a halo of NOD2 antibody around this chitin particle inside the cell. And this seems to be the pathway which is required for the uptake of chitin and then the activation of IL-10. And Jeanette did this in a whole series of ways. This information was published last year, so you can go and look at it. This is just one way. So she followed the chitin and co-localized chitin in the various receptors. And so this is a sort of mixed staining slide. And when you have green staining for chitin and red staining for the various receptor, you have a yellow stain. So here there are yellow stains. This means that the receptor and chitin are co-localizing. And this, for example, just to show you the logic here, this is a mannose receptor deficient mutant. You never see yellow staining inside the cell because the mannose receptor is required to transport the chitin particle into the cell. If you don't have, if you have a NOD2 deficient background, of course you never see yellow staining. You do see green staining because there's no receptor to bind. So that was the kind of logic. And then what Jeanette said was, well, if that's the case, okay, so we know how it gets in a bit. Does it make any difference to inflammation? So she did experiments in mice where she injected chitin into the peritoneum, and then she measured the inflammatory infiltrate, and she did that chitin plus or minus LPS. Now, as you know, LPS will induce a very positive inflammatory infiltrate, and if you follow the bars here, and again, the sun is following me around to the bars I want to show you, but in every case, I'll go down here, that if you have chitin plus LPS, you have a reduced inflammatory response. So the chitin was offsetting the inflammation you saw due to LPS. And she followed this over the course of a week, what you could see. So this is a 7-day experiment, and here's the pro-inflammatory signal going down, as evidenced here by TNF, and this is the anti-inflammatory cytokine going up. And this line at the bottom, this flat line, is a strain which is unable to do that, show that transition from TNF to IL-10, and that is a chitin deficient strain. So that transition requires chitin. The transition also requires chitinase, because it turns out that the chitin which is transported by the mannose receptor has to be just the right size to fit in the mannose receptor binding pocket. And if you block human chitinase, here's a human chitinase inhibitor, then you can block that response. But you don't block the induction of chitinase. Chitinase is not induced by chitin, it's actually induced by the mannan outer layer. And actually, to begin with, I thought that's confusing, but it makes perfect sense. That's exactly how you should design the cell, because there is no chitin presented normally to the immune system, but there is a lot of mannan. So mannan induces chitinase, and chitinase induction occurs even in the absence of chitin, but the production of the IL-10 response depends on transport of chitin into the cell via the mannose receptor. So what does that all mean? So now here's the arm-waving conclusion. So we wanted to explain this phenomenon which we didn't understand, why these mice their kidneys packed with candida, why do those mice survive? And here's one hypothesis, and it is that these high chitin cells, which had been exposed to Casper fungin, we know that they block pro-inflammatory signals and induce anti-inflammatory cytokines like IL-10. And if the pathology is due to inflammation, then you can offset. You have an immunotolerant situation in the kidney in this circumstance, where the chitin is offsetting the inflammation, which is otherwise due to the mannose-based recognition mechanisms. So that transition requires chitin. The transition also requires chitinase, because it turns out that the chitin which is transported by the mannose receptor has to be just the right size to fit in the mannose receptor binding pocket. And if you block human chitinase, here's a human chitinase inhibitor, then you can block that response. But you don't block the induction of chitinase. Chitinase is not induced by chitin, it's actually induced by the mannan outer layer. And actually, to begin with, I thought that's confusing, but it makes perfect sense. That's exactly how you should design the cell, because there is no chitin presented normally to the immune system, but there is a lot of mannan. So mannan induces chitinase, and chitinase induction occurs even in the absence of chitin, but the production of the IL-10 response depends on transport of chitin into the cell via the mannose receptor. So what does that all mean? So now here's the arm-waving conclusion. So we wanted to explain this phenomenon which we didn't understand,
why these mice packed, their kidneys packed with candida, why do those mice survive? And here's one hypothesis, and it is that these high chitin cells, which have been exposed to Casper fungin, we know that they block pro-inflammatory signals and induce anti-inflammatory cytokines like IL-10. And if the pathology is due to inflammation, then you can offset. You have an immunotolerant situation in the kidney in this circumstance, where the chitin is offsetting the inflammation, which is otherwise due to the mannose base recognition mechanisms. So, in a kind of scheme of things, what happens normally is the fungus detects its outer cell wall, the fungus is detected by its outer cell wall components, predominantly mannose containing sugars. This generates a pro-inflammatory response and pro-inflammatory cytokines, but it also, as part of that, primes things for the future by inducing a variety, in fact 2 major chitinases. These are actually one of the most dominant proteins which are induced by inflammatory neutrophils and macrophages. So chitinase is produced, the cells presumably are contained and killed off, and chitin then is generated, which is just the right size to engage with the mannose receptor, be transported into the cell, induce that signaling pathway, which leads to transcriptional activation of the genes encoding things like IL-10, and that switches off the inflammatory response. Now, a kind of, again, big way of thinking about this, sort of global way of thinking about this, is the way I think it sort of makes sense, is that chitin is the toughest molecule in nature, it's the last thing to go. Normally when chitin is exposed, probably the fungus is dead. You probably strip the wall to the bone, and it's probably dead. So, if you see a lot of chitin, it's usually probably indicating you've got the skeleton of the fungus, a dead fungus. And therefore it makes sense for that to be evolved as a signal to switch off the immunopathology which would otherwise result of having a sustained pro-inflammatory mechanism activating. The battle has been fought and won. At least that kind of makes sense. It gives us lots of experiments to do to go and try and prove that. And when we were doing this, there was a few other things done by completely different groups which kind of resonated there. Here's a paper, a mouse model paper, showing that this inflammatory disease, inflammatory bowel disease in this case, if you flood the lumen of the gut with colloidal pure chitin particles, you get IL-10 and amelioration of symptoms. Again, a completely different observation, entirely different research group. Here's some human polymorphisms which correlate with susceptibility to Crohn's disease, and they include mutations in TLR9 and NOD2, part of that signaling pathway, which is to do with anti-inflammatory pathway, which I pointed to. These seem to kind of fit, if you want. All right, so what have I told you? Here's the sort of take-home messages. I want you to remember that the fungal cell wall is a very tough structure, but it's also extremely flexible, and it's extremely mercurial and changing in its nature. It's a highly regulated structure, and it's probably safe to say that probably every single candida cell which has ever been generated by nature has a slightly different cell wall. So it's therefore quintessentially an extremely difficult target for the immune system, and also that really affects the way in which we see the way it will respond to antibiotics. And these mechanisms which protect it against antifungals, they're not new mechanisms. They've evolved in nature over many hundreds of millions of years, but they can significantly affect the MIC. So what you measure in an in vitro test might be quite a long way away from the actual MIC of a cell which has been exposed to a kind of candida in the human body for a few hours. And the fungal cell wall is composed of molecules which are amazing, and they're fungal specific. They're very important for activating the immune responses, both positively and negatively. And I guess they have the potential in the future to be therapeutic immunoactivators and immunomodulators, although that's something for another day. So the stars of this show I want to emphasize in particular, I showed you some early work by Jules Ennie, who worked with Al Brown, who's now left the country and is working in the US. But Jeanette Wagner is still at Aberdeen, and she's done this lovely work on chitin. There's been some very important collaborative works with people like Mihai Netia
Hector Mormont, who's now left, and the early work on chitin regulation continuing, being a wonderful collaboration with my colleague and former student, but is now a senior academic in her own right, Carol Monroe, many of you who you'll know. And here are some of the other collaborators. And this is, I think, our last party picture in December of the Aberdeen Fungal Group, or those who were not abroad at the time. And thank you very much for your attention.